Okay, good morning, everyone. Before starting, just a reminder for those of you attending online. So I remind you that you are on a webinar mode, which means that your mic and cameras are automatically switched off. If you want to ask questions to the speaker at the end of the talk, you can use the Q&A tab, which is on Zoom. And then we will allow you to switch on your camera and mic to ask the question. And if you are too shy, we can even ask the question for you if you put the question in the discussion box. So it's my pleasure today to introduce Nicolas Loiseau, who is a CNRS researcher at UMR MARBEC here in Montpellier. During his life before MARBEC, he has been working mostly on marine fish diversity in overseas territories, including New Caledonia, La Réunion, and French Polynesia, where he did his PhD at UMR EIO. Nicolas works on marine fish diversity, on all aspects of marine fish diversity, from taxonomy to functional traits. He combines theoretical approaches on the performances and the benefits of different diversity indicators, combining this with new technologies also on about monitoring diversity, such as autonomous underwater video sampling, environmental DNA, etc. And he applies this to the study of marine fish diversity, including, for instance, the spatial-temporal dynamics of reef fish communities and biodiversity conservation. So today he will present us some of his work on the effect of marine reserves on marine fish diversity. And Nicolas, the floor is yours.
Thank you, Bruno, for the invitations, and thank you, everyone, for being there. And so today I'm going to focus on the benefits and conservation paradox of MPA. So coastal marine fishes are highly diverse. There are many species around the world. They are central in the ecosystem functioning. They regulate over marine species populations, and they have also huge contributions to people. So it provides protein, macronutrients for billions of people. However, marine resources and mainly fish declining at an alarming rates. More than half of the oceans are under industrial fisheries, and most of the stock are overfished. So here you have the percentage of stocks that are sustainable in blue and unsustainable in brown. It's moving. It's not reacting. Okay. It's Eduoram. Eduoram is off. Great. I can. It's the same thing for you. Or? Yeah, it's the same. good, we're back. Okay. So in the same time, fish being ectothermic, so they are impacted by the increase of temperature by climate change, induced by climate change. Particularly by extreme events such as marine heat waves. So here you have an illustration of marine heat wave in front of Montpellier for this year. So each time you have the color in orange or red, it means that it's a marine heat waves. So it's quite scary. And in response of all these threats, marine protected area have been established to protect biodiversity and also to sustain fisheries. But only 8% of ocean are globally protected by MPA and only 2% by the take area, so very restricted protected area. And the main goal is for 2030 to have 30% of coverage of the ocean by protected area. So marine protected area and APA are widely recognized to be an effective tools of conservation. For example, the size, you have here the size and here the years of protections. Size of fish increase with the protections. We have also greater density, that biomass. So here you have here the high protected reserve, but influence the most the biomass of coral reef fishes at global scales. It's also well known that the highest trophic level are well protected by protected area. Here you have the infish and here's the fished area. And we see that the top predator are mainly in the infish area. And that's exactly the same things here. You have the top predator and here is the biomass in function of different level of protections. So the no entry is traditional, and here it's when it's very far for humans. And there are also recent papers that show that protected area could also mitigate the impact of climate change by increasing carbon sequestrations and coastal protections. And so it gives an adaptive potential for society and also for fish. However, there is still lots of issues. The first one is positioning of MPA. It's a trade-off between fisheries, politics, socioeconomics constraints, and at the end, diversity. It's a difference, the difference with terrestrial MPA that are established to protect diversity. Here it's mainly to provide protein for human populations. The second point, it's also mainly due to the first point and the trade-off between politics and fisheries. MPA are small and medium to limit, small or medium size, to limit the conflict of user of the maritime space. There is also a lack of regulations if there is no control in the MPA. The MPA cannot be effective. I will give you some numbers that speak for themselves. Here it's for Mediterranean Sea, and for 72% of MPA we have a lack of regulations. So that means that fishermen can fish in the protected area, and we have only 0.2% of protected area, but with the highest effective level where all human activity are forbidden. And the last point is the limited international networks. Once again, still linked to the first point, the trade-off between fisheries and politics. The management of MPA networks is done at national and local scales. There is few managements at larger scales, at international scales. So we rarely take into account the network of MPA at global scale. So it's a strong issue since fish are moving between protected area. So there are still some questions. For example, the first one is the extent to which MPA support different facets of biodiversity, taxonomic, functional, genetic diversity. The extent to which MPA are well connected and able to effectively seed other MPAs. Extent to which MPA support evolutionary potential of fish populations, so have a higher genetic diversity inside. How to integrate the ecosystem functioning and process into a conservation strategy. And the last point is deeper water can be a natural protected area. So I will discuss this 5 points during the presentations. I'm going to start with the extent to which MPAs and protected area in general support different facets of biodiversity. So for this point, I will talk about birds, fish, and plants. It will be the early times. We work with it's a meta-analysis where we look at the difference in diversity and in compositions between strictly protected area and the less protected neighbor in a buffer of 50 kilometers. So for birds, for plants, and fish. We can expect different results. The first one here, the big circle is a protected area and the small circle is a non-protected area. So we can expect that the non-protected area hosts less species and is a subset of the protected area. We could also have, for example, this kind of patterns where both non-protected area and protected area host the same number of species, but the composition is completely different. So it's called beta diversity. And we have all the intermediate assumptions between the turnover and messiness. So the SPA is strictly protected area, NPA is non-protected area, and RA is restricted area. It's protected, but human activity still occur in this place. And first, we'll look at the difference in number of species between protected area and then non-protected area neighbor. And we find that generally, especially for fish and birds, that the number of species between protected area and non-protected area doesn't change. It's very similar. The delta is close to 0. And for plants, we found that protected area hosts less species than the non-protected area. For plants, it can be easily explained by the fact that we work with data sets from the Alps and protected area are at the top of the mountain. So there is a huge natural selections and only a small subset of the diversity can live there. And then we compare the composition, the species compositions between these different types of management. So here it's the beta diversity between strictly protected area and non-protected area. Here it's strictly protected and restricted, and here it's restricted and non-protected. In green, this is the beta diversity. So here's the composition is very different. And the two facets of the beta diversity, the turnover, so the replacement by a species by another one, or the nest and nest, it's just a difference in species diversity. Is one of the assemblages is a subset of the other one. And as you see, the beta diversity is super strong. It's around, for fish, for example, it's around 0.5, so 50% of species are different between the protected area and the non-protected area. And we find exactly the same patterns between the different taxa. We found, for example, that between 12% and 50% of species occur only in non-protected area. So that means protected area missed completely some species. And for threatened species, only 58% only occur in strictly protected area, and we have very less value for plant. For example, it's only 7% of threatened species of plant that live only in protected area. So several process can explain the heterogeneity in species compositions between protected area and non-protected area. First, the very sensitive species may maybe provide refuge in protected area, and they become too rare to be detected outside the protected area, so it increases the beta diversity. Some species enjoy the human activity, like the pigeon, for example. So it increases the heterogeneity of the species composition. Protected area are generally small, so they cannot capture the whole range of all species. So at the end, all this process increase heterogeneity of the biodiversity, and it maximizes the regional diversity. And the last point, it's important to note that the richness is not a good proxy of the effectiveness of protected area. It's not a good indicator if protected area is effective or not. After we found this result with the meta-analysis, we decide to test it on the field. So we sample fish communities in Mediterranean Sea with eDNA, and we sample inside the protected area, 5 kilometers outside and 10 kilometers outside. And we found exactly the same result that we found in the meta-analysis. So it was great. So here it's the overall species richness inside MPA, 5 kilometers outside and 10 kilometers outside. So there is no big difference. Here it's a predict species richness. So it's when you consider it's output of a model that controls for the difference in habitat and environmental conditions. So when you control this effect, species diversity is even higher outside the protected area. And once again, we found that the turnover, so the replacement bias of a species by another one is super strong between protected area and outside. And the funny part is we look at which kind of species influence these results. And we find that the cryptobenthics, the very small fish that live on the substrate, have a higher diversity outside the protected area and create this beta diversity. And our hypothesis is the number of predators are higher in the protected area. And so by predation, they reduce the presence of this kind of species in the protected area. So after this discussion about diversity and species compositions, I will focus on to the extent of marine, to which marine reserves are interconnected and have also benefits at a long distance. So why connectivity is important for MPA? Fish are moving a lot. They have some lava space where the fish drift with the current. Then there is times where the fish is looking for a favorable habitat to stay. So it's essential to have this overview of the connectivity between site and protected area to have an effective management. And it's mainly important because the local impact on the MPA may have a huge impact at distant place. For example, here you have an MPA with lots of spillover of fishes, but if it's MPA is not connected with this one, all the benefit of the first MPA is lost. And MPA may have an important contributions on the connectivity and on the gene flow via the active dispersal of individuals that live inside the MPA. Also with passive dispersal with the currents, the spillover of individuals and larvae. And there is also benefits with, for example, translocations in Indonesia. They're working a lot with translocations. They take some fish from MPA to seeds over MPA. So it's not a natural gene flow, but it's one of the benefits of MPA to increase the connectivity between sites. So our questions was, is long distance benefits of marine reserve is a myth or reality? So on this map in color, it's all the MPA at global scale, and the color is the nearest neighbor distance. So when you are in yellow, you have another MPA close to you between 0 and 40 kilometers. And if you have this blue color, that means you are far from every other MPA. And we look at the average dispersions of marine species in the literature. And we found that in average, marine species disperse at 42 kilometers. And when we compare the nearest neighbor distance between MPA and the average dispersions, we find that most of the MPA could be connected without regarding the current. So it seems that's good news. But the biggest MPA, so with the best benefit, are not well connected. Here you have the size of the MPA, and here is the nearest neighbor, and all the big MPA are far from other MPA. So they are not able to seed other MPA. After this paper, second paper show that finally, when we consider the current and the larval dispersal, MPA at the end are not well connected. So it's based on only on model of current and larval dispersal. And they show that at global scale, MPA are weakly connected. With the current, only 42% are not seeded by any other MPA. We have 2% that are completely isolated, and the big one and the most effective one. And 53% of MPA do not receive any larvae. And the last point is the country with the highest dependency to coastal fisheries receive very little larval supplies from MPA. So there is a gap in conservation strategy to create effective network. So the first paper was a review. The second paper considers the current, and after this work, the idea is to what about the reality when we go on the field and you look at the connectivity between sites. So during the reserve benefit project, it's led by Stephanie Manel. We sample more than 1,000 fish from 4 species along a large gradient on the west part of the Mediterranean Sea, and there is 8 protected areas in this sampling. And we look at the relationship between the relatedness, genetic relatedness, how close are individuals, and the geographical spatial scales. So here for each point, sorry, it's not so clear, but for each point, it's the value of the slope of the relations between relatedness and spatial distance. And the dark points are the significant slope, and if you have no dark point, it means that the slope it's not significant. So there is a very, very restricted dispersals. For the Serranus, the most dispersal individuals, at 30 kilometers, after 30 kilometers, it's quite impossible to find a genetic related individuals. But during the project, we find two very close individuals between two MPA, and they were separate from more close to 300 kilometers. So that means even if it's very rare and there is a restricted dispersal, some MPA may have a huge impact on the connectivity at large scales. Still on the same project, MPA are often assumed to conserve the evolutionary potential of marine populations, to conserve the genetic diversity of fish. So we use exactly the same individuals from the same sampling. And the idea was to compare the genetic diversity of the 4 species between the 8 protected area and the outside of the protected area. So each individual was genotypes at more than 10,000 SNPs. So here it's a multivariate analysis. So each point is a fish for the 4 species. The circle is inside the MPA, the triangle is outside, and the color is the closest protected area. And from this multivariate analysis and models, we found that only environmental variables, so salinity, temperature, and chlorophyll, influence the genetic diversity, so genomic variations between individuals. But protected area are not detectable effect on the genetic diversity. I can show you more on this figure. Here it's the genetic diversity versus inside and outside MPA for the 4 species. And we find an homogeneous patterns of local genetic diversity within an outside reserve. Only the Serranus cabria show a lower genetic diversity in protected area. Could be a kind of paradox, conservation paradox once again, where the diversity is lower inside the protected area. But it's yeah, the pattern it's very, very small. We could explain these homogeneous patterns between protected area and non-protected area by the fact that MPR may be too young, so we need time to have a diversification of the genetic, and also too small to protect the overall genetics. But it's important to have this kind of multi-species approach to inform the conservation planning in general. This kind of analysis focus only one species, and it's nice to have an overview with different species with different traits. After diversity and genetics, I'm gonna discuss the need to integrate ecosystem functioning and process into the conservation strategy. So in marine protected area, traditionally standing biomass is the indicator of the effectiveness of marine reserve, and it gives you information of the state of the reef ecosystem. But it don't only give you information of how much fish is present at the moment. And it did not give you any information about the productivity and the flux in the assemblage or the site of the reef. So we need to integrate the biomass production, the effectiveness of the biomass productions in the conservations and management. And it's particularly important because it gives you also information on the recovery capacity of your site. So we work with the Reef Life Survey dataset. It's volunteer divers that sample close to 2,000 tropical reefs. They count close to 1,400 species, and we record 4 millions of individuals, and they also record the size of each individual. So it's a huge data set. So thanks to the diver, we have the size and abundance. So with the size and abundance, we can compute the biomass in fish of each site. And then for each individuals, we predict the growth rates. And so we were able to compute the biomass productions, also considering the natural mortality of fish. So we have the standing biomass, the biomass productions when we consider the growth rates of the fish, and the biomass turnover, which is a ratio between biomass productions and standing biomass. And it gives you information about the effectiveness of the biomass production. It's a renewal of the biomass 4 times. So we compute these metrics for all sites. And you have here, for each point is a site. Here it's the standing biomass, and here is the biomass turnover. And we classified this reef in function of these 2 metrics. So here in green, where we have the site with the very high biomass. On dark blue, we have site with a medium and low biomass, and also a large biomass, but with a strong biomass turnover, a strong effectiveness of the biomass productions. And here we have the site where the biomass productions is very low, and the standing biomass is also very low. So this kind of classes give you a nice information about the management of this reef. So we look at, for each site of these classes, we look at the structure of the trophic food web. So here we have the site with the high biomass, it's a high turnover site, and here it's a low biomass turnover site. And we found that top predators are present in the high biomass with a lot of also herbivores. And for the low biomass and low turnover, we don't have any top predator, and we have very few herbivores, and it's mainly detritivore. And for the high turnover sites, it's we have a lot of turnover and a lot of herbivore, and that could explain why the biomass productivity is so effective, because this kind of fish grow faster, so give you more biomass grow times on the reef. Then we model these categories with human and environmental variables. So you have here the 3 main drivers. This one is a primary productivity of the ecosystem. Here it's a human gravity, so it's kind of human impact on the site. And here it's a temperature. So we show that the site with the very high biomass are far from human activities. But when we look at the site where the turnover, the biomass turnover is strong, we have impact of the human populations. So we can explain that, but the fact that human fish, when the fisheries increase, the size of the fish decrease, and it's like, and the smaller fish grow faster. It's like human baby when we are young, they grow faster, and so forth. So to finish at the end on this category, it's conceptual because we work at global scales, and the conservation is more focused at local scale. But the exercise gives you some nice overview of the state of the reef and to adapt the management. Where the biomass is very strong and the productivity is low, that means you need to protect this reef, because they are the last reef with a relatively high biomass. You cannot fish on this site. But on this site where the productivity is strong, you can adapt the fisheries and the management in function of the context. If your sites are in a very rich country with a small dependency to marine resources, you can protect these sites to go through the high biomass site. But if you are from a country where the marine dependency is strong and you need the fish protein, you can just adapt the management at a traditional management and ban industrial fisheries and to have output for human society. And this kind of site with low biomass and low productivity, it's kind of conservation traps. It's high pain for low gain. So the conservation output for this kind of site will take a lot of times. So after that, it's difficult to discuss with people who say, okay, the protected area is not working, we have no more fish. I will finish this presentation with a natural protected area or not. Is deep water a refugia? So Mesophotic reef system, it's a part of the oceans between 30 meters and 150 meters. And in general, these sites have a lower stressors, such as climate change, pollutions, and also fisheries. So the hypothesis from several papers is that deep water could buffer the impact of the bigger stressors, and so be a natural MPA. So considering the multifaceted of diversity, we try to test this hypothesis of the refugia. Here it's taxonomic refugia. You are a strong taxonomic refugia when you're here, and here you are a strong functional refugia. The dark blue is mesophotic assemblage, and the clear blue is shallow assemblage. The circle is for taxonomy, and the square is for functional. So we can expect several results. For example, mesophotic area is not a refugia. It means that both shallow and mesophotic do not share any species in common and any function in common. Refugia could be a functional refugia. The two, the shallow and the deeper water, do not share the same species, but they share the similar traits. We can have the assumptions E, where the mesophotic area is a complete refugia for shallow water, because both share exactly the same species and compositions. And we have also the we cannot see, but the last hypothesis, it's the super refugia, where the mesophotic hosts the same species, the same traits, and have also unique species and unique traits. So we sample sites in Mayotte using a video system from 8 meters to 100 meters. The camera records diversity, abundance, and we have 5 functional traits for each species. So how we work, we use a framework based on hill numbers to have exactly the same framework of analysis for taxonomic and functional diversity. So for taxonomic, we compute taxonomic richness and taxonomy entropy that considers the abundance. And the same thing for functional diversity. With Q equal O, we compute the functional richness and also the functional entropy, but consider the biomass. First, we found that the biomass decreased with the death. So it was the first results. And we found similar results for alpha diversity. So here you have alpha diversity and here the beta diversity, so the dissimilarity in compositions and traits for taxonomic and for functional. So we show that the taxonomic diversity and the taxonomic entropy decrease with the death. And the beta diversity was super strong between the site of the shallow reef and the deeper reef. So it means the species composition is very different for the different depth. But for functional diversity, we found that the richness and the functional entropy do not change a lot between the shallow and deeper water. The beta diversity the functional beta diversity was also low, but the functional entropy beta diversity was strong. That means the trait compositions were quite similar between shallow and deeper water, but the distributions of biomass within the functional entities change a lot. I will illustrate this result. Here there is the functional space, so each point is a species, is a functional entity, sorry. And the size of the circle is the biomass. The gray form, it's the overall functional space for all record. And for its functional space, it's between 0 and 20 meters, 20 meters, 40 meters, 40 meters to 60 meters, and over 60 meters. And the functional space is quite similar between the different range depth, but the distributions in abundance change a lot. From here, where there are lots of herbivores, from here, where there are less herbivores and more carnivorous species. So at the end, deep reef cannot be a natural marine protected area. Despite we find similar functional trait compositions, there is a very limited species similarity in the species compositions. And that's been only a very small number of shallow species may be able to live on deeper water and vice versa. But the important message is when we extend marine protected area to the mesophotic reefs, we will maximize the protections of both species and functional diversity, and so increase the protection of the reef functioning. To conclude, MPA a fundamental role and unique role to protect unique species compositions and unique trait compositions. A mosaic of protections may maximize the regional diversity. It's not always a goal in conservations because you can increase the gamma diversity with the worst species. Once again, you don't want to protect the pigeon. So increasing the number of pigeons at the regional scale, it's not the main goal, but it's important to have this view of the impact of the mosaic of management. MPA poorly conserve the evolutionary potential of fish populations because mainly MPA are young and not big enough to enclose all the genetic diversity. So MPA needs to be bigger and more connected to increase the gene flow between protected area. MPA must include deeper water. For the moment, most of the coastal, most of the MPA are very, very coastal. So we need to increase the protections of deeper water. When it's possible, conservation strategy need to focus on the ecosystem functioning and to include process like biomass productions, reproductions, the productions of eggs inside the conservation management. So having this big picture is crucial. And fish conservation is multi-taxon, multi-function, multi-process, and multi-scale. It's very important to also focus at the very local scales where the threats are and where the context can influence a lot the output and the effectiveness of the protections. And at the end, it's multi-people. So all this work it's with amazing people work on the field, on the paper, on the model. So thank you.
Thank you very much, Nicolas. Do we have questions? The room. Yeah.
Thank you very much. It was very interesting.
Yeah.
Perfect. So a lot of the data that you presented today are based on comparing both kind of areas, protected versus non-protected. And just to make sure I understand, when you do that, do you compare all the data you have on protected versus non-protected area, or do you also take into account the characteristic of the areas, like the location, the kind of fisheries are around, these kind of things because it's just 2%, right, of the ocean that are protected. So how can you take this into account when you do your comparison? And I have just a second part of the question.
Okay.
Oh, you want to answer, because it's really linked. So
okay.
Because you had this model where you predicted the richness, for example,
yeah.
And in this one, you said that you took into account some characteristics of the non-protected area, and there you expected more richness than what you observe in your data. So can you then infer that you have a lack of richness in your data in this non-protected area compared to your model, compared to what you expect?
For all the works we control, but the influence of environmental driver and the context of the MPA. In the model, we have the size of the MPA. We have also age of the MPA when it's possible, but it's very hard to have this information. We have the fisheries impact, and we always control by the difference in sampling size, because in the model, we control the number of sample inside the MPA versus the sample outside the MPA. So we bootstrap the data to always have exactly the same sampling effort. Yeah, if we don't do that, for sure you have a huge difference between outside and inside. But yeah, and for the first example on the meta-analysis for birds, plants, and fish, we also model this difference in species composition in function of the difference in habitat, and actually it was not very significant. It was significant only for plants, once again because the MPA for plants was the top, and there is a kind of also in the restricted area, it helps they cut the tree in some parts to increase the diversity, because when the wood it's a completely forest, the diversity decreases a lot and you lost some. So there is a kind of also management of it. In this kind, for this taxa, the habitat influence a lot our results.
So we now have a question online by Mohammad Hassan.
Yeah.
Yes, we can hear you.
Okay. Thanks a lot for this presentation. Indeed, biological invasions in the Mediterranean Sea have already been shown to have triggered biogeographic modifications, range shifts of native species, population declines, or local extinctions. In fact, some alien species are occasionally the only ones that can sustain ecosystem functions in front of the environmental alterations imposed by global change. Considering this point, how it's possible to create MPA? Thanks.
So the question is how invasive species gonna change the management of a protected area, right?
Yeah, that's.
Actually, I don't know. We start working with invasive species. I'm working a lot with Sébastien Villéger on some, the invasion of modernity with the rabbit fish.
Yeah.
So actually, I don't know, because invasive species are targeted by fishermen too. So it's quite hard, but I mean, protecting the big predator inside the MPA, I think it's a good thing, because it could have a good influence by regulating the population of invasive species. But yeah, we'll see in the future.
Thanks.
Thanks for your talk. At the very beginning, you said that there is a small proportion of MPAs that are actually like controlled and like the regular, like with like really, you're sure that it's actually protected and not just on the paper. And so I was wondering, in your analysis, if you take all the marine protected areas or just the one that we are protected, because otherwise maybe it's not necessary to have bigger MPAs, but just real MPAs, you know. So.
Yeah, for the meta-analysis, we make the difference between the very strictly protected area. That's why there is a strictly and the restricted. The restricted is where you have still human activity is managed. For example, you exclude some kind of fisheries activity, industrial activities, but you still have fishermen. So we split this different management. But the same thing for the paper of Raphael on the productivity. We have different classes of protected area, where we have some protected area with a real effectiveness, and the protected area with small regulations. But if we start to work only on protected area, we have a huge control. Yeah. Yeah, we can see. I mean, you can still see for the compositions on this one, you have here it's a difference between strictly protected area and non-protected area, and here it's a difference between strictly protected and restricted. So we saw that the difference in species composition is lower, but between restricted, strictly and non-protected. So yes, there is an influence, and there are some work from Josh Sinner that show that it's the control of the protected area. It's one of the main driver that influence the biomass.
So now a question online by Philippe Borsa.
Yes, thank you, Nicolas, for your talk. My question was, it emerged suddenly when you were talking about transplantations of fishes in Indonesia to enhance connectivity, gene flow. Well, first, MPAs, I'm not aware of any MPA in Indonesia, which is a real MPA. There is a lot of fishing and a lot of illegal things in Indonesian MPAs. But my point is, why would artificially enhancing gene flow be a desirable thing for species that may well be adapted to their local conditions? So without knowledge of this, transplanting fishes from one MPA to another, it may not be a really good idea.
Yeah, I totally agree. Yeah. But I mean, the expectations behind this translocation, it's when you increase the gene flow, you increase the probability to have an adapted gene to climate change or other threats. So the hypothesis behind this activity to increase the probability of the fish to adapt to future conditions. But yeah, I'm agree with you. It's kind of weird activity to translocate fish.
Yes, well, especially in the Indonesian setting, when we look at maps of phylogeographic maps of that region for fishes or giant clams or anything, we find a high structure, very sharp structure caused perhaps by I don't know, reproductive isolation, unsuspected cryptic species or whatever. And so to play the sorcerer is no good even for adapting to changing conditions. I mean, let the local populations adapt their way as much as possible.
Yeah, yeah, I agree.
Other questions?
Thank you for the talk. I was just wondering, in the study where you looked at where you sampled in the 8 MPAs in the Mediterranean, if they were fully no-take MPAs.
Yeah, they were fully no takes. It was quite hard because the idea it was not to kill fish, so we catch them during dive night, and we catch them, take a small piece of the fish, and let it. So yeah, it was completely no take area.
Thank you.
So we now have a question online by Cécile Luc.
Oui, bonjour, merci beaucoup. Thank you very much for the presentation. One time we were talking about human impacts, and I was wondering if it could be interesting to compare the different sources of pollution along a coast and see the impacts on diversity and productivity. Because, for example, we can measure concentrations of metals, pesticides, or other pharmaceutical products, and we can know the toxicological impacts on fish. So yeah, I was wondering if it can help to see the diversity and productivity.
Yeah, I totally agree. But the problem is to have this kind of, we work at the global scale, so having a precise information at the scale of the pollutions and nutrients inputs from human activities was quite impossible. But since we work with the human gravity, and it can be seen as a proxy of the anthropogenic impact for each site. But yeah, totally agree. If we work at local scales, it's important to have this kind of information to compute productivity.
Okay, thank you.
Thank you, Nicolas, for your presentation. I have one question of MPAs. Your experience or your study. What is the main driver of MPA effectiveness? Because we are our time interest after the establishment of MPA to test if it is really effective about the conservation of marine biodiversity, about the recovery. So according to you, what are the main driver of MPA's effectiveness after its establishment? And yeah, maybe I can let you answer about the question first. So I have another one, but if I'm okay with this first question, I can ask this again.
It's control and size.
What?
Control and size. If you have a big MPA that's well managed with some control for policemen, and it's effective.
So for you, it's about the well enforcement of the MPA. So.
Yeah, the well enforcement. It also depends of some country and then socioeconomic context, but there are also traditionally managed protected area, mainly in New Caledonia or French Polynesia, and it works super well because it's very well managed by people that know the sea, that know the resource, and that manage, and they know what they need. So in general, it's worked very well. But for the Mediterranean Sea, it's just a matter of size and control.
So if you establish an MPAs, so it's not relevant to talk about his. If the MPA is not well enforced, it's not relevant to talk about its effectiveness.
Yeah. If there is lots of MPA on the paper, and it's only on the paper, and it's a huge problem for protected area. For example, when you extract, there is a data set of all MPA at global scale called WDPA or something like that. And when you plot all the MPA, you have a nice map and say, okay, it's well covered. But when you reduce and filter, but real MPA with local management by traditional management or management like in Europe with some enforcement by police or etc. It's very small.
Okay. So after the establishment of MPA, we have to be sure that the MPA is well enforced. And do you think that the duration of protection is also in effect in a main driver because in a many study we found that if you have survey or monitoring the MPAs after 2 years after establishment, the effect you will see or the effect you will measure about the effectiveness of MPAs is not relevant because we don't see the effect of protection that can be related to other things. I don't know, but it's not really the effect of protection that you are going to measure. What is your I don't know, your opinion about that? Is it relevant to take into account also the duration of protection and say, okay, we have to wait, for example, we have to wait after 5 years before to measure any effectiveness of this MPA? Is not relevant. What do you think about it?
I think it's context dependent. There are some papers that show you need more than 10 years before measuring the effectiveness of your protected area. But it also depends on the context of the MPA. If you start protecting a nice place with already nice compositions and high biomass, it will be fast. But if you start to protecting a devastated area, it will take more time. But it takes time. After 2 years, I think it's quite impossible to have an MPA.
Or online. Last question.
Okay, well, thank you for the for the presentation. That was very nice with a lot of results and discussion. I have a question regarding to the potential project or studies tackling the transition waters in conservation. I mean, the brackish transition water seems to be very important ecologically meaning for conservation because they host all the larvae and other kind of ecological functions. Is there any study? Is there anything on that? Because in your presentation, that was almost all on strictly marine protected area, but well, that was the subject, but well.
Yeah, I don't know well the subject. The only things I know it's when you build an MPA that integrates all this kind of ecosystem and even the land ecosystems with marine and you have an overview of all, you protect everything and you have nice results. But yeah, I don't know about this.
Could be very interesting to compare the MPA that holds this kind of ecosystem you just talk about, and the other one that just have, I don't know, maybe the marine area ineffectiveness.
Some result with the mangroves, where we show that the protected area that host mangroves ecosystem and also coral reefs ecosystems are more effective because all the juveniles and larvae are in the mangroves. So it protects the exchange and the gene flow between the two ecosystems. And yeah, for sure it will be nice. Yeah, but maybe some people work on it. But
okay, well, I will be interested in getting these papers. Thank you.
Thank you again, Nicolas. We will close the session now. So thanks a lot.